Good day, and thank you for standing by. Welcome to the Roivant Sciences fourth quarter and fiscal year 2022 earnings call. At this time, all participants are in listen-only mode. After the speaker's presentation, there'll be a question-and-answer session. To ask a question during this session, you'll need to press star 11 on your telephone. You will then hear an automated message advising you your hand is raised. To withdraw your question, please press star 11 again. Please be advised that today's conference is being recorded. I would now like to hand the conference over to your speaker today, Stephanie Lee with Roivant. Your line is open.
Good morning, and thank you for joining today's call to review Roivant's financial results from the company's fourth quarter and fiscal year ended March 31st, 2023. I'm Stephanie Lee with Roivant Sciences. Presenting today, we have Matt Klein, CEO of Roivant. For those dialing in via conference call, you can find the slides being presented today, as well as the press release announcing these updates on our IR website at www.investor.roivant.com. We'll also be providing the current slide numbers as we present to help you follow along. I'd like to remind you that we'll be making certain forward-looking statements during today's presentation. We strongly encourage you to review the information that we filed with the SEC for more information regarding these forward-looking statements and related risks and uncertainties. And with that, I'll turn it over to Matt.
Thank you, Steph, and thank you everybody for listening this morning. It's great to be back. I was talking to the team this morning and saying it feels like a slightly anticlimactic call because we were obviously just together last week to talk about the really exciting data from the chronic period of our RVT-3101 study. But actually, an enormous amount has happened for us, both in this fiscal year generally as well as specifically in recent quarters. So looking forward to providing updates on those topics. We'll talk a little bit about where we are through the course of our year. We'll give some great updates on the progress that we're making with the ongoing launch of VTAMA, as well as a reminder of our atopic dermatitis results. We'll do a quick refresh of the data we put out last week on RVT-3101, a quick update on our FcRn program, a financial update, and then we'll turn the line over to Q&A. Starting on slide 5, just kind of as a reminder or a level setting, look, we're really proud of the continued progress that we've made here. We celebrated this quarter with ADORING 1, the 10th consecutive positive phase 3 study that we have run. That's the most recent 10 studies that we've run have been successful. We now have 6 products that we've gotten approved by FDA out of our model. We reported $1.7 billion in cash as of March 31st, which supports our cash runway into the second half of 2025, before which we'll have a tremendous amount of data to share. And then we are incredibly proud of what we now believe is an industry-leading pipeline, especially in late-stage I&I, with over $15 billion of sales potential, supported by the ongoing launch of VTAMA and with a number of potential best or first-in-class programs. We've said all along on slide 6 that 2023 was going to be our biggest year. We are right at the midway point, both in terms of the year and in terms of the data that we've been looking forward to sharing. We've continued to provide updates on VTAMA, as we will do today, and this has been another great quarter of progress for that launch. We've now shared data for both of our phase 3 studies in atopic dermatitis for VTAMA, data that we think is really, really exciting and will support an important product in that class pending potential FDA approval next year. We've now supported data from both the induction and chronic periods of our study of RVT-3101, our anti-TL1A antibody in ulcerative colitis, which again is phenomenal data. We think sort of really top-end efficacy that has the potential to really matter for patients and to be an important new option. So we'll continue to provide updates on that program. Still coming, and obviously closely watched, are a number of updates from our anti-FcRn franchise, including the healthy volunteer study for IMVT-1402, which we hope and believe will establish that program as a best-in-class anti-FcRn antibody, as well as a number of ongoing trials in batoclimab to show continued efficacy of that agent and the class in multiple indications. And then finally, at the fourth quarter of this year, and we will talk more about this before it comes, we have our readout of brepocitinib, our TYK2 JAK1. It's a potentially pivotal one of 2 studies in SLE, which we think has the potential to be, again, transformational top-level efficacy in that patient population. And on slide 7, just as a reminder, we are just very proud of our portfolio, of our pipeline overall here, with a number of late-stage agents that we think matter in some of the biggest classes, certainly in immunology, and We will continue to add to this pipeline as opportunities present themselves. Obviously, we made some really important additions within the last 12 months with IMVT-1402 and RVT-3101, and I hope we can find more just like those two to bring on in the coming year. So I will start in the next section here with an update on the commercial launch of VTAMA. On slide 9, I'll say we are very excited about the way that demand continues to evolve for VTAMA. We are the best launching novel topical, we believe, in psoriasis history. We are obviously the best-selling branded topical in psoriasis and have been since very shortly after our launch. We continue to like the way that the demand has grown, and we expect to see it continue to build through the end of this year as various things, including coverage and our DTC efforts, build. On slide 10, some really great progress here in the early launch that we just wanted to make sure we hit. First of all, we did $13.7 million in net product revenue for the March 31st quarter, which is up a pretty good degree from $9 million or $9.2 million in the prior quarter. So obviously happy with the sales number. Maybe, frankly, even happier with the progress that we made during the quarter in gross net yield, up from 18% to 25%, which is a reflection of the breadth of coverage that we added, frankly, faster than expected in the first quarter of this year, and we've continued to add coverage since. So, we're really happy with that. In some ways, I think we managed to pull forward gross to net improvements earlier this year, and we continue to feel good about our trajectory through the year. We'll talk more about that over time. From a coverage perspective, on slide 11, we're now up to 76% of commercial lives covered within a year of launch. This is better by a meaningful margin than our expectation at the time we launched the product. This includes the addition of two national PBM formularies, 2 national health plan formularies during this period, an important regional PBM formulary with 18 PBS plans. Just really great coverage. And as a reminder, the significant majority of that coverage is single step through steroids, which is exactly where we want it to be when we launch the product, and which gives us access to the patient population that matters most to us. As a reminder, there are almost 400,000 topical corticosteroid scripts every week between psoriasis and atopic dermatitis. And, we're currently doing about 4,000 or a little bit more than 4,000 scripts a week. So we have a ton of room to grow into that opportunity. And we're really excited to do that both in psoriasis today and pending FDA approval in atopic dermatitis as well. Speaking of atopic dermatitis, on slide 12 here, just a little reminder. We've talked a lot about this data. We got together to talk about the ADORING 2 data on a call in March. We put out the ADORING 1 data as well, which was extremely consistent. This is it's great data. We are really excited about this. It's data that is really, it's terrific on vIGA response rates, really, really good EASI-75s, and I'm very happy with the quality of the itch data here. The reported data was in adults. It looked very good in children as well, and about equivalent. And, we feel like in AD, which is really a disease marked by itch, these data are going to make a really big difference for patients. And as a reminder, this study went all the way down to pediatric patients at age 2, which is great because the pediatric patient population in AD is large and the unmet need there is significant. Across all patients, and especially in pediatric patients on slide 13, one thing that you really care about with a topical agent is safety. And I think it's clear from our data that the tolerability of this agent in atopic dermatitis is very, very good. Frankly, even a little bit better than we saw in psoriasis, with very low rates of contact dermatitis, very low rates of follicular events, just a clean profile that gives us exactly the profile as a product we think will matter to physicians and patients. So really excited about this data from a safety perspective as well. I won't spend too much time on the cross-trial comparison on slide 14, but this is data in a moderate to severe patient population that looks competitive with or better even a number of systemic agents, and certainly in the same ballpark as or better than anything else that is a topical in atopic dermatitis. So more updates to share on Dermavant over the course of the year. Looking forward to continued coverage, looking forward to progress in the franchise, looking forward to providing updates on the sNDA filing for VTAMA AD, which we expect to be at the very beginning of next calendar year. Looking forward to sharing those on quarters to come. So I'm going to pivot now and give a brief reminder. This is data that we put out just last week, so I won't spend a lot of time on it, with RVT-3101. On slide 16, we really believe this program is in a class of its own, right? Anti-TL1A antibodies, at this point, between our program and one of our competitors that also reported data earlier this year, this is a remarkable class of drugs that has shown incredible efficacy, and we are very proud to be the first agent to show sort of real, sort of proper blinded 52-week data last week. And it's great data. We showed substantial improvements between week 14 and week 56 across basically all of the endpoints in our go-forward dose. We are the only anti-TL1A antibody at this point with validating long-term efficacy data, and we have over 200 patients of such data. We have a biomarker that we've talked a bit about that is relevant in 60% of the UC population with meaningful improvement in efficacy even relative to the all-comers population. And then safety has been very good for the agent in the study so far, and we've seen no impact of immunogenicity on the program, which we know was a question that people were hoping to get run to ground in this trial. So we're now full speed ahead here with a plan to run a simple phase 3 program with a single subcutaneous dose in the near future, and we'll provide updates on that just after our discussion with FDA, which is coming this summer. As a reminder of the data, and I'll go quickly on this on slide 17. We saw modified Mayo clinical remission. This is in our phase 3 dose, go from 29% at 14 weeks to 36% at 56 weeks. A remarkable 50% at week 56 in endoscopic improvement. And then endoscopic remission, this is an endoscopy sort of zero, is something that frankly very few agents hit, and so we don't typically see it reported. And we were really excited what we saw there with 21% of patients in the all-comers population on the phase 3 dose achieving a clear endoscopy. Once you overlay the biomarker on slide 18, that data looks even better. It gets up to 43% clinical remission in the phase 3 dose at week 56, 64% of patients meeting the bar for endoscopic improvement, and again, a really exciting 36% of patients in endoscopic remission by week 56, which, look, we didn't measure specific antifibrotic markers here, but personally, I look at this data as suggestive that we are having a potential disease-modifying impact on these patients, so really exciting. Another topic, and this is also data that we shared, but just as a reminder, in a large patient population here of really the whole study, in patients who are biologic experienced, with our biomarker, we saw, frankly, just phenomenal data at 56 weeks, 34% clinical remission for biologic experienced patients, 45% endoscopic improvement. This is some of the best data that's been shown in biologic experienced patients, which are generally recalcitrant and hard to treat. And as a reminder, this includes patients on doses other than our optimally chosen dose. So we expect when we generate large N data in our phase 3 study that there's room for improvement even over these really great numbers. So very excited about the potential for efficacy in the biologic experienced population. I think any way that we look at our data where there's sufficient N to reach a judgment, we feel very confident that this is going to be a great agent in later lines of therapy. Obviously, all that has to be backed up by a good safety profile. And one of the really exciting things about the TL1A class, and you can see our safety data on slide 20, is because of the way TL1A works as a mechanism, because it is really only sort of present in diseased inflamed tissue, it's sort of a signal amplifier. You don't get some of the infection and other things that you see in other anti-inflammatory classes. And so we had no severe infections observed and no infections observed at greater than or equal to 5% rate in the chronic period. And just generally, we saw a really good safety profile through week 56, well tolerated at all doses, and serious AEs were sporadic and determined by the sponsor not to be related to drugs. So very, very clean safety profile that we think is going to help the utility of the drug going forward. And then finally, on the data on slide 21, as a reminder, one of the questions that we got a lot going into the maintenance period was whether immunogenicity was going to matter. We were reasonably confident that it was not. What you can see on this slide in the chart in the middle, this is across the data pooled across all 9 arms of the study. Patients with ADAs, by and large, had somewhat better clinical remission than patients without ADAs, and it was uncorrelated with the quartile of ADA titer. We don't think this is a real effect, obviously. We just think this is noise. We think that ADAs are having no impact on safety or efficacy across all the arms of the study. And we had said we expected our neutralizing antibody rate to be flat to down. I'm proud to say it was 0 at week 56 in the expected phase 3 dose. So we had no patients with neutralizing antibodies at week 56 on that arm. On slide 22, without belaboring the point too much, we feel really great about the amount of data that we have here, and it is quite differentiated relative to the field here. We have over 400 subjects dosed including 250 patients dosed across an IV and 3 subcutaneous doses, 200 patients dosed across 3 subcutaneous doses out to a year of dosing, just hundreds of patients dosed no matter how you cut it. We are the only agent here with subcutaneous data, the only agent here with anything like this quantum of long-term data. Our biomarker, which was prospectively specified, has obviously been tested in many patients, and we expect a commercial form factor that is a once-monthly subcu auto-injector. So we really feel great about our profile versus the other agents in this class, which also have what we expect will show promising data given the quality of the target. Finally, on slide 22, I wanted to highlight we have initiated, as we mentioned last week, a phase 2 study in Crohn's disease. It's just over 100 patients. It is 2 doses subcutaneous monthly, similar in that sense to the phase 2b study that we just ran in UC. There'll be 12 weeks of dosing followed by a 40-week chronic period, again, similar with a similar set of endpoints, which you can see on slide 22. This was a study that we wanted to start very quickly because we realized as we took a step back that we felt like we could run a proper dose-ranging study in Crohn's patients without giving up any ground on the opportunity to be first in class in Crohn's. And that would allow us to run, just as we are doing in UC, a straightforward, simple, single-dose phase 3 study that will be optimal for patients. One question I expect we'll get is why no placebo arm in this study? The basic answer to that is we want to get the dose-ranging study done as quickly as we possibly could, and so we wanted to make this a really attractive study for patients to enroll in starting now. And so we're excited to have that study up and running. So overall, as a reminder on slide 24, just really excited about this program. Think it is a major anchor in our late-stage pipeline. We think it will be the first-in-class anti-TL1A antibody with an efficient, well-validated path to approval on a single dose carried forward to phase 3. We think we are uniquely positioned to overcome some of the limitations of IBD therapies, including efficacy in later-line therapy with sustained clinical remission and endoscopic improvements among the highest ever reported. We think our biomarker further differentiates this class and our agent versus other treatment options for IBD patients and gives us an opportunity to select for patients with an even higher clinical remission rate, although notably we think our data clearly supports a study in an all-comers population irrespective of biomarker status or line of therapy, and our expectation is that that is the patient population that we will be targeting for approval. And then finally, just many opportunities for additional growth, including the Crohn's study I just mentioned, as well as the dual targeting of both inflammatory fibrotic pathways, opening up a range of large markets and high-end need indications that go well beyond IBD, and we're looking forward to sharing more on that just as soon as we're underway with additional studies. Finally, I want to spend a few minutes on IMVT-1402 and our anti-FcRn franchise. Obviously, Immunovant has spoken about this program, and there's no new updates in this section, but just wanted to remind everybody of what was coming, given how excited we are about this program. So on slide 26, as a reminder, we have a proper franchise of anti-FcRn antibodies with our first generation batoclimab currently in multiple pivotal studies across MG, TED, and CIDP, with data coming over the next couple of years in those studies as well as others. And then we have our next generation antibody, IMVT-1402, which we believe will have the same best-in-class suppression of IgG as batoclimab, while showing minimal impact on albumin and LDL, and therefore being useful for chronic dosing and diseases with an even larger population. We have data coming from that program in August, September for single ascending dose data, and October, November for multiple ascending dose data. And we think that has the potential to establish that agent as best in class. As a reminder, both of these agents are proper classic subQs. They are subcutaneously administered. They are simple subQ injections. They will be sort of at-home administered, we believe, and sort of very straightforward, comparable to other subQ programs on the market, which we think is also, at this point, something differentiated and something we do not believe any of the other agents in the class have today. Our phase 1 study that's ongoing in IMVT-1402 has, as I said, single ascending and multiple ascending dose cohorts. That study is underway, and we are looking forward to sharing that data later this year, as I mentioned. We believe that the data that we have in hand is likely. We have NHP data that we've shared on these calls and other forums. And based on our data for batoclimab, both on albumin and on IgG. We think the SAD data, which we will be putting out, as I said, at the end of the summer, may be usefully predictive of MAD data. I get a fair number of questions on the bar for success. And I guess the one thing I'd like to say is, look, we believe based on the NHP data and based on the way these programs are engineered that we should be clean on albumin and LDL. Remember that the LDL assay has about a 10% variability and the albumin assay is a little bit more specific than that, but has some variability. And frankly, although we have seen, to be clear, none of this data today, so I have no information about what this will show, and our expectation from the NHP data is that it ought to be clean. Our advisors tell us that even up to, for example, a 10% excursion in LDL would not be clinically meaningful. So looking forward to sharing that data, optimistic given the way that drug was engineered as well as the NHP data that we should have a good result there, and looking forward to getting back together with the Immunovant team later this year to go through it. Finally, I'll give a brief financial update on slide 21, and then we'll open the line for Q&A. So, we showed net revenue of $27 million, including net product revenue of $14 million for the quarter ending March 31st, or net revenue of about $60 million, and net product revenue of about $28 million for the fiscal year. For the quarter, we had R&D expense of $130 million, or adjusted R&D expense about $126 million. And SG&A was 126 or adjusted about 100. So, sort of consistent with prior quarters from a spend and burn perspective. And notably, we ended the fiscal year with $1.7 billion in cash equivalents, which we feel good about as far as carrying us into that second half of 2025 guidance and giving us lots of opportunity to turn over data cards. So look, I won't go through all of the catalysts on slide 33. I'll just say we've talked about some of them here, but there is a long list to come of important developments, including in programs we've talked very little about, frankly. And so we're looking forward to sharing that data, looking forward to getting back together. It's been a tremendous fiscal year for us. It's hard to believe this is only the second 10K we filed. I want to thank all of our patients and the team at Roivant, as well as those listening on this call for being with us. And with that, I will pause and turn the line over to Q&A. Thank you, everybody.
Thank you. As a reminder, to ask a question, please press star 11 on your telephone and wait for your name to be announced. To withdraw your question, please press star 11 again. Please stand by while we compile the Q&A roster. Our first question comes from Robyn Karnauskas from Truist Securities. Your line is open.
Hi, guys. Thank you for taking my question. So just to going back, Matt, on your comments around the bar for 1402, do you think, given the properties of the molecule, that we should look at the graphs that you've shown for the SAD and MAD data and it will look similar? Are the properties of molecules similar enough that we should expect the curves to look similar? That's my first question, then I have a follow-up.
So I'll also invite Frank if he has any comments there. And obviously, the Immunovant team talks about this often. I'd say overall, in terms of the broad properties, I think the answer is we would expect them to look generally similar. Obviously, with the notable difference that 1402 has been engineered to avoid the albumin binding, or the albumin interference, steric hindrance, and so I would not expect to see an impact on albumin and LDL. Frank, anything you'd add to that?
I think that's well said. The only thing I'd add is they are not identical antibodies, obviously, so they have slightly different properties. They are similar. I would encourage the investment community to kind of look at the albumin data as probably the most robust and reproducible assay among the assays we're going to be presenting in the future. But I do think that the curve should be directionally similar.
And for IgG as well. That's what I was really asking. And then a question on Vtama. So, congratulations on doing a great job selling so much drug this quarter. I was just curious, can you just give us some trends on what you're seeing and feedback from the remittent effect? At what point might you expect to see patients not taking the drug or pausing the drug and then waiting for the disease to come back a little bit? I know it's early in the launch, so just wanted to see if you're seeing anything there. And that's it. Thanks.
Good. Thanks, Robyn. It's a great question. And I'll say broadly, first of all, physician and patient feedback on the remittive benefit is great. I think it's something that patients experience very quickly on drug, right? People go on, they use it for a while, they go off, they see the effect preserve, and we get a lot of feedback from docs and from patients that they are excited about it. I suspect that is embedded in the renewal rate for the product already. That is like the rate at which patients are renewing, the number of tubes that they're using is a function of that already. It's obviously a little bit too early to tell how that shakes out versus the overall growth and demand. But in general, it's not like I would expect to see some sort of significant change in the future because of that effect. And I think it's a selling point for the drug. I think it's something that physicians are happy to tell their patients that they can go off drug and continue to see a benefit. In terms of tubes, I guess I'm not sure that was exactly your question, but our view has always been that in order to be successful, we need to be a steroid replacement, and that steroids sort of just over a tube a year, maybe a tube and a quarter a year. It already looks from our data like we are in excess of that. It's hard to tell how much in excess of that, given how early it is, but my expectation is that we will have more tubes per year than steroids, mostly given the lack of a duration limit on the drug.
Great. Thank you.
Thanks, Robin. Really appreciate the question.
Okay, one moment for our next question. We have a question from David Risinger from SVB Securities. Your line is open.
Thanks very much. So congrats to you, Matt, and your team for all the nice updates and the corporate progress. So with respect to my questions, I guess I'll start with VTAMA. So the trends are obviously moving in the right direction, but on slide 9, it does show that the TRX peaked. And I know that there are sometimes anomalies in the data, but the TRX peaked for a few weeks, close to 5,000, and they're not back at that level yet. So could you just talk about the trajectory ahead and the commercial drivers for the continued ramp of the product? And then second, with respect to the approval clock for AD, once you file it in the first quarter of 2024, how many months after filing would you expect approval? And then finally, a higher level question, could you talk about second half 2023 transaction opportunities? So should we expect potential acquisition of additional external assets? Is it possible for Roivant to monetize an undervalued asset? Any framework for thinking about potential external transaction activities would be helpful. Thank you.
Thanks for listening, David. Those are all great questions. I will take them in order. First of all, on the script trends, thanks for asking the question. Look, I think first of all, there's clearly just like week-on-week variability in these scripts, and that's been true for all of the topical launches. It's not just ours. And so I think a little bit of the sort of quote-unquote choppiness is noise, and I expect we will continue to break through the ceilings that we set for ourselves over time. It may not be linear. The particular peak that you pointed out was just on the back of AAD, and I think there were some docs who were maybe trying the product out on the back of that first atopic dermatitis data. And I guess my hope and expectation is that that's a preview of what's to come when the AD approval comes in terms of the level of enthusiasm around that data. But obviously the product is currently not approved in that indication. In terms of timelines, so it's an sNDA. We expect a 10-month approval clock is the short answer after filing. And then in terms of transactions in the second half of the year, look, I think it is clear from our history that we are not great at sitting still. It's a pretty exciting market in a lot of different directions. Obviously, some of the targets that we are in are targets that are closely watched. They're targets that big pharma companies clearly care about. So you can imagine those are discussions that we're thinking through economically and trying to make sure we do the right thing for patients and the right thing for each program in turn. And then in the other direction, it remains a very fertile opportunity for new programs. Big pharma companies are constantly making portfolio prioritization decisions. There's obviously the patent cliff dynamics and other things facing the industry that have provided us opportunity in the past still obtain. And I think you can expect that we are looking actively at lots of things. And I hope to be able to share some of them with you over the course of the rest of the year.
Great. Thank you.
Thank you, Dave.
Thank you. Our next question comes from Neena Bitritto-Garg with Citi. Your line is open.
Oops, sorry. If you can hear me. Thanks for taking my question. Just wanted to ask a follow-up question to Robin's question earlier about what you're looking to see with 1402. Just in terms of the albumin reduction, since it sounds like that is the most reliable sort of assay to look at, how should we think about what an acceptable albumin reduction looks like? I believe with the lower dose of batoclimab, or I think the 340 mg dose, you saw about a 30% reduction, which corresponded to about a 40% increase in LDL. So I guess, is there a benchmark on albumin reduction that we should be thinking about in terms of translatability to that kind of 10% or less LDL increase? Thanks.
Yeah, thanks, Nina. And I'll ask Frank if he has any comments as well. But look, I think the short answer is lower is clearly better, and our expectation is it will be categorically different than what we saw with batoclimab. They're much lower. I think the sensitivity of these assays is in the kind of mid-single-digit range, and so I would say anything within that range should be completely acceptable as noise and will, in our view, lead to very low LDL in any reasonable patient population. So that's probably how I answer that question. Frank, anything you'd add to that?
No, I think that's well said.
Sounds like no. Perfect.
Okay.
Great.
That's super helpful. Thank
Appreciate
you.
the question.
One moment for our next question. We have a question from Corinne Jenkins with Goldman Sachs. Your line is open.
Yeah, thanks. Good morning. Maybe a couple of questions from us. First, what can you share with respect to fill rate and the time from prescription to fill for VTAMA? And it sounds like it's early, but whatever other color you could provide on refill rate at this point would be helpful. Maybe I'll start there and ask my second one as a follow-up.
Thanks, Corinne. Appreciate the question. And I'll ask if Frank has anything to add here. I think our general view is patients getting scripts filled has not been a significant challenge at this stage. Early in our launch especially, we were heavily seeing fills through specialty retail derm pharmacies, which are a direct channel that the dermatologists work with all the time, and that we're very knowledgeable about VTAMA, knowledgeable about our copay card program, and so on. So I think that was helpful. At this point, with our insurance coverage, frankly, many patients who show up at the pharmacy are just covered at the pharmacy. And so again, fill is as easy as any other product at Walgreens or CVS and so on. So I think we're not seeing much in terms of challenges getting scripts filled. On refill rate, look, I think we've been happy to see growth in NRX as well as good growth in TRX, and I think we're seeing a good number of refills. That's, in our view, a measure of the fact that patients on drug are happy to be on drug and continue to use it. And again, we don't have that duration limit. So I don't have much to add beyond the data other than say I think it's clear from our current refill rate, as I said before, that we will be in excess of steroids in terms of tubes per year. Frank, anything you'd say on either of those points?
I think the only thing to add is as you know, our coverage has been increasing meaningfully over time, and that is there's a little bit of a lagging effect, obviously, in how that coverage comes into play and finds its way into downstream plans. And so I think you'll see our the ease of prescriptions at the point of sale only continue to get better as that coverage has come and continues to come into place.
Okay, helpful. And then as you think about, I think you referenced direct-to-consumer campaigns and spend, just how do you think about balancing that spend versus the coverage and yield rates you're working with today, and how can that change over the course of the year?
Yeah, thanks. It's a great question, and obviously, and I think we've talked about this in various forms before. Early in the launch, when we didn't have the kind of payer coverage that we do now, DTC is a real balancing act because you can definitely drive volume through DTC, but if you're not getting coverage, you're driving volume that is not sort of commercially helpful volume per se, and it does cut against GTM. Where we are now with 76% commercial coverage, I'd say in general, demand generation is going to be helpful. And so we've increased sort of the picture around DTC. The Dermavant team has put together a great campaign. It's out there now, and we're sort of still in the early days of it, figuring out exactly the right targeting plans and sort of getting real-time feedback from the marketplace. but feel good about what that's going to mean. I would expect to see that take some time to filter into the script trajectories. It takes a number of impressions before patients respond to DTC and come into the office and so on. So, that's something to watch for kind of through the end of this year and beyond. But in general, I think it will matter. And then, in terms of spend, I'll just say it's included in the various guidance that we've given in terms of where we expect it to be. And I'd say in the grand scheme of things, it'll be well worth it for the demand that we generate on the back of it.
Great. Thank you.
Thank you.
Thank you. Thanks for your questions.
One moment. We have a question from Dennis Ding with Jefferies. Your line is open.
Hi. Great. Thank you so much for taking our questions. Just two for me on TL1A. For your Crohn's program, thanks for the diagram in your deck. Can you comment on if you will use similar doses as you did in UC, or could you explore higher doses? And maybe talk a little bit more about the biomarkers. Is this the same biomarker from UC? And then number two, I saw in your 10K that for the TL1A, 20% of patients who are biomarker positive did not give consent. Can you confirm if that's true and maybe give a little bit more color as to why and perhaps the commercial implications there? Thank you.
Perfect. Thanks. So on the dose question, I'd say we're pulling from a similar range of doses in the Crohn's study as in UC. So I think that's the answer on dose. And on biomarker, we are using the same biomarker algorithm in Crohn's as we used in UC, and our expectation is that the patient population should be similar, the size of the patient population. And our belief, based on our understanding of our biomarker algorithm, is that it should obtain equally Crohn's. Obviously, we're excited to see that data from the study. On your question about the 20% consent, so this is something we had shared at the time of the induction data. Basically, the biomarker samples were gathered from all patients in the study, but the analysis of those samples required a separate consent, and the sponsor of the study did not obtain consent for 20% of the patients in the study for those samples to be analyzed because of local IRB and site policies. So basically, all of our biomarker data, biomarker positive and biomarker negative, effectively reflects data on 80% of the study population, whereas all comers includes even those 20% of patients for which the biomarker data was not gathered. We have no reason to believe there's any relationship between those consents and severity or anything else. The data are robust. There's plenty of N, but that's just the way the study was conducted.
Great. Thank you. And maybe as a quick follow-up around the LNP patent litigation, what's going on there, and when can we get the next update? Are we still waiting for that claims construction order, maybe in 2024? Thank you.
Yeah, thanks. So, those cases, and there's several of them at this point, are ongoing. And we continue to be pleased with the overall progress now that we're actually in the meat of the case. We are in the discovery process in the Moderna case now. And so you can imagine new information will be forthcoming that will affect the progression of the case. But most importantly, we're gearing up for that same claim construction hearing and claim construction order that will come at the beginning of next year. That will probably be the biggest, most publicly visible next step, although plenty will be happening behind the scenes over the course of the fall.
Thank you.
Thank you.
Thank you. And one moment for our next question. We have a question from Yaron Werber from TD Cowen. Your line is open.
Good morning. This is Joyce on for Yaron. Thanks for taking our question. Maybe just a follow-up on the question before about 3101 in Crohn's. Is there a hypothesis here that it might work better in Crohn's, just given that the fibrotic component is a bit more involved in this disease versus UC? And then you obviously showed really great data on endoscopic remission in the phase 2b UC study. Can you just maybe comment on what you're hoping to see there in Crohn's disease and maybe what other studies have shown in the past on this endpoint? Thank you.
Yeah, perfect. Thank you. These are great questions. Look, obviously, the study will show what it will show. Certainly, the line of questioning you're asking is one that we've thought of too. That Crohn's is an even more fibrosis is even more involved in Crohn's disease than it is in UC. And when we look at things like the specific markers of fibrosis that were identified in Pfizer's phase 2A study, as well as the endoscopic remission and the clean endoscopy that we saw in our own phase 2B in the chronic period, I think there's certainly reason to believe that we could potentially be even more differentially efficacious in CD. And look, agents typically that work in one typically work for the other. So, I think that's the other sort of generally historically true fact, and certainly the biology of TL1A suggests the same would be true here. And then, look, our biomarker, as I just said, also we think should carry through into CD and should give us an opportunity for differential efficacy in a large subset of CD patients as well. In terms of endoscopic remission specifically, it's a good question. I don't have a super specific answer. I think Crohn's patients may have more tissue involvement than UC patients, so that may be a slightly tougher endpoint in that patient population. But on the other hand, we're delivering an antifibrotic benefit that could matter, and I would say it certainly seems plausible that we would see benefit in endoscopic remission as well.
Okay, great. Thank you.
Thank you. And our next question comes from Brian Cheng with JP Morgan. Your line is open.
Good morning, guys, and congrats on the progress, and thanks for taking my question this morning. Maybe just first one on TL1A, related to your phase 2 trial design that you have ongoing for Crohn's. I'm just curious if you can provide a bit more color on the dosing that you have selected, specifically in the chronic period. It seems that you will only be evaluating one specific maintenance dose, based on slide 23. Just curious if you already have a sense of what the dose will be for the chronic portion. If not, what would be the deciding factors for the chronic portion? And I have a follow-up. Thank you.
Yeah, thanks, Brian. Look, appreciate the question. We believe we got a fair amount of information from the dose ranging in our UC study that has been helpful in making a prediction here, a pretty robust understanding from our dose response there that's given us a good deal of confidence in Crohn's dosing. And we are pretty confident that we know what our chronic period dose will be in the Crohn's study. So there's not really any ambiguity for us in that. We are, for the same reason that we have not shared specific dosing information in UC, we're not going to share our choice of doses at this stage for the CD study. We will be happy to share those around the same time we're willing to share dose ranging data in UC next year. But in general, dose response that has been identified in UC is generally predictive across IBD, and so we feel pretty confident in our selection of doses there. As a reminder, we have very robust dose ranging data at this point for 3101.
Okay. I guess related to Vants, which I don't think a lot of investors have looked at yet. Just curious, one is, how should we think about the timing for this update, given that you will have Vant data spread through the fall, and also you'll have prursentinib data in the fourth quarter. So one is, how should we think about the timing from the phase 1/2? And then also, what should we think about the expectation for the lower risk MDS data? How do you think about the bar there, for you to move forward to the next stage?
Yeah, perfect. Thanks, Brian. It's a great question. You're absolutely right. We don't get it very often. So the study for 2001 is an open label study, and we've had good progress on enrollment. So we're going to have a decent number of patients worth of data. We will provide the update probably pretty late this year, frankly, because the longer we wait, the more data we have, and I think we want to provide an update on the most comprehensive set of that data. I think in terms of the bar, as I think we'd said before, in the earlier study that AZI had conducted, or the earlier portion of the study that AZI had conducted, there were about 20 patients, or a little bit less than 20 patients, that had the sort of profile that we are looking for here, that were sort of low-risk transfusion-dependent MDS patients. And we saw sort of a little bit higher than 30% transfusion independence rate. Look, I think what we're looking for here as we move into earlier line patients is something that would be sort of obviously competitive with or superior to luspatercept, which, as I'm sure you know, is in kind of the mid-40s in patients without prior lenalidomide exposure. So I think that's probably the first thing. The second thing we're looking for is we are hoping that a decent number of these patients will have this biomarker aberrant TMM14C transcripts, where we saw a very high response rate in the earlier patients. And if that's true, we should be able to provide some update on how we are doing in that patient population and whether that opens up a precision oncology strategy. So that's broadly what we're looking for here. We will share that update, as I said, very late this year, almost certainly after the
Thanks, Matt. Great. Thank you, Matt.
Thank you. One moment for our next question. We have a question from Louise Chen from Cantor. Your line is open.
Hi. Congratulations on the quarter, and thanks for taking my questions. So I wanted to ask you, if you're approved for atopic dermatitis, is all the work or the payer work that went into VTAMA for psoriasis, can that be leveraged? And then secondly, on rheumatoid arthritis, is that a potential opportunity for Immunovant? And if so, what do you think your go-forward plan would be there? And then lastly, just wanted to ask you for brepocitinib, big data readout coming at the end of the year. What do you think you need to see to move forward with this opportunity, and why do you think you'll be successful when others have failed here? Thank you.
Yeah, thanks, Louise. Those are all good questions that span the portfolio, so I'll take them in turn, but thanks for asking them, and thanks for listening, as always. So on the first one, the short answer to that question is definitely yes. That is, all of the payer contracting work in psoriasis is extremely helpful in atopic dermatitis. Obviously, at some level, there is an additional sort of formulary positioning question that needs to be answered because the cascade of therapies in AD is slightly different. But the contracting work, all of the commercial work in psoriasis will translate and be very helpful in getting quickly onto formulary in AD. In terms of 1402, look, the sky's the limit from an indication perspective. And I think, especially now that we think we have really the only true at-home, easy sub-Q, any indication is eligible. RA, it's an interesting question biologically. It's probably not something that we were initially very focused on. J&J has this ongoing study that they've talked a bit about, and they've said they are going to share that data later this year. You can imagine in autoantibody RA patients that there could be an opportunity. And I'd say if the J&J RA data looks good, we could be very, very quickly in, frankly, a pivotal study if we needed to, and compete with them, probably, again, in sort of the more severe patient population. And I think that could be a really, really exciting opportunity for 1402. And given 1402's profile, the simple sub-Q, what we believe will be the cleanliness and albumin and LDL, and the depth of IgG suppression, we think that's an example of an indication where if J&J's data is good, we think we ought clearly to be the best-in-class agent, and frankly could be neck and neck for first-in-class at that time as well. So really exciting possibility. On brepocitinib and SLE, we've talked a little bit about this. It's not a simple numerical bar. Look, I think it's about building on the data that we've already seen, including data in baricitinib as a JAK1, more recent data in upadacitinib as a JAK1, and then the data that we've seen in deucravacitinib as a TYK2, combined with the knowledge that brepocitinib, which has now been in well over 1,000 patients, has outperformed in cross-trial comparisons both JAK1s and TYK2s across many, many, many studies. Remember, it's been run in 5 large late-stage sort of phase 2 studies at this point. And so, look, I think that combination gives us optimism that the agent could perform very well in SLE. the other factor with SLE always, and you mentioned some of the challenges others have faced, is these studies are difficult to conduct. Pfizer has the conduct of the study, although we've had a lot of input, and I think we're doing everything we can to ensure, including the study learned from many of the prior designs, to optimize for the possibility for success there.
Thank you.
Thank you.
Okay, our next question comes from Douglas Tsao with H.C. Wainwright. Your line is open. Douglas, please check your mute button.
Oh, sorry, I was on mute. Can you hear me now?
Yes.
Hey, Matt, thanks for taking the questions and congrats on the progress. Just given what we saw from Tuscany, obviously with 3101, you're starting the study in Crohn's. I'm just curious how much more broad, or how are you thinking about the opportunity with that asset outside of IBD? So obviously Crohn's is a natural sort of first step, but are you thinking about other indications in I&I with that asset? Thank you.
Yeah, thanks. Look, the short answer is yes. We are thinking about opportunities that go broader to other inflammatory disease, to other fibrotic disease. I think given the quality of the data in IBD, the playing field is very large. And so we're thinking about ways right now to narrow it down a little bit and figure out what our best lanes are. And we're looking forward to sharing more of that color as soon as we can. Look, there's just a lot of biology to support indication expansion here. And frankly, TL1A knocks down so many different targets along the inflammatory and fibrotic axes that you can look at, like we have an impact on IL-6. You can look where the IL-6s are. We have an impact on TNF. You can look at all the many places the TNFs are. So there's just a lot of different places to go. And I think our goal is to be smart about it, to find things that benefit from the full axis, to find things that fit from a commercial perspective together with where we're starting, and to try and learn as much as we can from small studies to guide our decisions. So that's all sort of to come soon. And
I guess, Matt, as a follow-up, within the portfolio, I&I has certainly become a clear focus from a business development standpoint. Obviously, bringing in 3101, you brought in brepocitinib. I'm just curious, at this point with the portfolio, how are you thinking about the balance between bringing in new additional assets as they potentially might become available versus pursuing some of the natural expansion opportunities, or sort of just running new studies in different indications than the initial targets? Thank you.
Yeah, look, I think on the one hand, we are very pleased with the coherence of our late-stage portfolio and of the fact that we've built an I&I pipeline that we think is among the best out there. And so I think we're proud of that. And we think insofar as opportunity begets opportunity, we will continue to get calls and look at lots of different things within I&I. That said, when we started talking about the TL1A program with Pfizer, 1402 hadn't yet presented itself, and we obviously didn't have TL1A yet, and we still had, I think at the time, we still had our sickle cell program, and we weren't obviously an I&I company. And I think if we had said we're a something else company, we might not have had the opportunity to work on TL1A. So I guess what I'd say is the next TL1A, if it is outside of I&I but that attractive, is still something we will work on. So look, I think we see lots of opportunity in lots of places. We like the concentration, but not so much that it would stop us from working on something else great in another area. And we see plenty of exciting things inside and outside of I&I.
Okay, great. Thank you so much.
Thank you.
Thank you. Appreciate the question.
I would now like to turn the conference back to Mr. Matthew Gline for any closing remarks.
Yeah, thank you, operator, and thank you everybody for dialing in this morning and for listening. I know we made you do 2 calls in the space of a week, so we appreciate it. We have a lot more to share over the course of this year. Look forward to getting back together later in the summer. So thank you again, and have a great day.
This concludes today's conference call. Thank you for participating. You may now disconnect.